Welcome to the MedWire News podcast. In this edition, we are discussing the Phase III SUMIT trial, which investigated Selumetinib, a MEK inhibitor, in the metastatic uveal melanoma setting. The results were recently reported in the Journal of Clinical Oncology, and we spoke to study author Paul Nathan from the Mount Vernon Cancer Centre in Middlesex, UK.
This was a trial in metastatic uveal melanoma with Selumetinib, a MEK inhibitor, in combination with dacarbazine compared to dacarbazine chemotherapy alone. And the reason we did it was because my colleague Rich Carvajal in New York performed a study with Selumetinib on its own in a local randomized study compared to dacarbazine on its own. And he found significant benefit in favor of Selumetinib with a hazard ratio of just under 0.5, with a doubling effectively of PFS. So there was some academic interest that there was some evidence of activity with Selumetinib in this disease before we embarked upon this trial.
So there was a robust rationale for undertaking this trial, but what were the results?
So in this randomized trial, a total of 129 patients were randomized to receive either Selumetinib plus dacarbazine or dacarbazine on its own in a 3-to-1 ratio. With the primary objective of progression-free survival. And unfortunately, the hazard ratio for PFS was 0.78, with no statistically significant difference in PFS between the standard arm of dacarbazine and the experimental arm of dacarbazine plus Selumetinib. So there was not a significant improvement in PFS compared with dacarbazine. There are a number of potential possibilities as to why the findings were negative. The most obvious one is that Selumetinib does not have significant clinical activity in this disease. However, we do have a positive Phase II trial of single-agent Selumetinib, which showed activity in uveal melanoma. And there is a biological rationale because we know that MAP kinase pathway is very frequently activated in this disease. So another possibility could be that the combination with dacarbazine somehow means that the potential benefits of Selumetinib are not manifested. And there is not good in vitro evidence looking at the combination with dacarbazine and Selumetinib, whereas there is evidence for other cytotoxic chemotherapy combinations with Selumetinib as having anti-cancer activity.
It's not uncommon for the results of Phase II and Phase III trials to be discrepant. Positive Phase II results are often followed by negative findings in the Phase III stage, more often perhaps than researchers would anticipate. But why is this the case? What are the reasons for the differences?
Yes, it's a very interesting question. There are multiple reasons for that. Often Phase II studies are performed in limited numbers of centres with specialist interest in the disease, and therefore one finds that the patients are very highly selected, whereas more often in larger randomised studies you have a more representative disease population being examined of the disease as a whole rather than a selected group of patients. And there are confounders, for instance, that the fittest patients will travel to a specialist centre, but the less fit patients, or those with more aggressive biology, will tend not to travel to a specialist centre. And so if your trial, with an early phase clinical trial, you tend to have them performed at specialist centres, but later phase studies will look at all centres, and so the case mix is different. So there are potentially subtle differences between patients. This trial had central review of imaging, and although we did see a difference between investigator interpretation of scans and central review of imaging, it doesn't explain these negative results. Finally, there's just a sample error effect. So the more patients you treat, the more robust your observation. And if you're treating very small numbers of patients, or comparatively small numbers of patients, you're more likely going to get a spurious positive or indeed a spurious negative answer.
The next question is, what do these negative results mean for Selumetinib in uveal melanoma? Is it the end of the road for the drug? Dr. Nathan doesn't think so. He explains that there are options that remain to be investigated, including looking at different combinations and different schedules.
We are currently performing a clinical trial looking at an alternative combination of selumetinib in combination with paclitaxel. It's a 3-arm study called CELPAC with 13 sites in the UK, and we're just about to open a 14th site in Berlin. And the design of that study is to investigate the activity of selumetinib in uveal melanoma, either with selumetinib on its own or in combination with paclitaxel. And there's a scheduling difference in the 2 chemotherapy-containing arms of the study. There's also continuing interest in selumetinib in the States. My colleague, Dr. Carvajal, is performing a study with the agent in uveal melanoma looking at a discontinuation regime. We've got some evidence from the laboratory that intermittent exposure of uveal melanoma cells to MEK inhibitors may result in improved benefit. So that's being tested in a current clinical trial under his care.
And what about the drug class as a whole? Would the results perhaps have been different with a different MEK inhibitor?
There are differences in the biological properties of differing MEK inhibitors. Selumetinib is not the most potent MEK inhibitor, but actually there is little clinical evidence that shows what's likely to be more significant benefit with more potent MEK inhibitors in uveal melanoma. However, there have never been any head-to-head comparisons. I think it's unlikely that alternative MEK inhibitors are going to be particularly attractive as investigational agents on their own for this disease. However, there's great clinical need for improvements, and there's a lot of work going on, not just with MEK inhibitors, but also with newer immunotherapeutics in uveal melanoma. And that's where a lot of hope is currently for a major improvement in outcomes.
We also asked Dr. Nathan about BRAF inhibitors. Recently, these have shown a lot of promise in patients with cutaneous melanoma, especially in combination with MEK inhibitors, and we wonder, is such a combination possible in uveal melanoma?
So uveal melanoma is very different from cutaneous melanoma, apart from the fact that they're both melanocytic neoplasms. So cutaneous melanoma has a high rate of mutations in BRAF, between 40 to 50% of cases do, and we know that BRAF inhibitors on their own or in combination with a MEK inhibitor are profoundly active for cutaneous melanoma. One almost never sees a mutation in the BRAF gene in uveal melanoma. One sees in greater than 80% and probably more cases than that activating mutations in 2 G proteins, GNAQ and GNA11, in uveal melanoma. And those do signal down a number of pathways, including the MAP kinase pathway, and result in signaling through MEK, which is why there was a rationale to examine a MEK inhibitor in this disease. But the disease is very different from cutaneous melanoma. One of the other areas where there's a major difference between uveal melanoma and cutaneous melanoma is in the action of immune checkpoint inhibitors. As you know, CTLA-4 and PD-1 checkpoint inhibitors are a standard of care in cutaneous melanoma, but they have very limited activity in uveal melanoma. And so we're looking at alternative immunotherapeutics in uveal melanoma because conventional checkpoint inhibition is not clinically useful in the vast majority of patients.
Finally, the SUMIT trial may have had a negative outcome, but the endeavour wasn't all negative. There were definitely some positives, as Dr. Nathan outlines.
One thing I would say is that doing trials in rare cancers is very difficult because these patients, there are not a lot of these patients around, and what it requires is really strong networking and collaboration between the clinical teams and their referrers, and also between the clinical teams running the study and sponsors. And the fact that we were able to deliver an international randomized trial in advanced uveal melanoma, which has an inherently very poor prognosis, I think is a testament to the degree of cooperation between the investigators on this study, and I think they should be commended for that. And it also bodes well for the delivery of future studies, which is the only way that we're going to be able to move treatment for this disease forward and improve outcomes for patients.
And with that, we'll sign off until next time. We thank Dr. Nathan for taking the time to speak with us and all of you for tuning in.